Hello everyone, in this lecture we are going to study acid-fast staining. There are some microorganisms that are not easily stained by simple staining procedure because they have limited permeability to stain solutions due to presence of waxy substances like macrolic acid in their cell wall. If somehow they get stained, they are difficult to decolorize even by strong decolorizing agents like acid alcohol. Such organisms are called as acid-fast organisms. The technique was developed by Paul Ehrlich in 1882-83 during his study on tuberculosis. Acid-fast staining is a differential staining technique. It differentiates the organism into two groups as acid-fast organisms and non-acid-fast organisms. Gill-Nielsen's method of acid-fast staining is most popular method. Definitions: acid-fast organisms: the organisms that get stained by acid-fast staining but do not get decolorized even by strong decolorizing agents like acid alcohol are called as acid-fast organisms. For example, Mycobacterium or Nocardia. Non-acid-fast organisms: the organisms that get easily stained by simple staining procedure as well as decolorized easily by strong decolorizing agents are called as non-acid-fast organisms. Procedure of Gill-Nielsen's method: take a clean grease-free slide. Prepare smear on a slide, air dry and heat fix. Flood the smear with Gill-Nielsen's carbalbuxin. The steaming is done for 5 minutes by placing the slide on water bath. Do not allow the stain to evaporate and dry. Wash the slide with water. Decolorize the smear with 20% sulfuric acid solution or 3% HCl and 95% alcohol mixture. Wash the slide with water. Flood the smear with counterstain, that is 0.2% methylene blue or 1% malachite green for 20 to 30 seconds. Wash the slide with water. Air dry the slide and observe the slide under oil immersion lens. Reagents used in acid-fast staining: primary stain is ZnCF stain. Decolorizing agent is 20% sulfuric acid or 3% HCl and 95% alcohol. Counterstain is 0.3% methylene blue or 1% malachite green. Mechanism: acid fastness in Mycobacterium or Nocardia is due to presence of high mycolic acid in the cell wall. Due to high lipid content, cells are less permeable to stain solutions. To penetrate the primary stain, two methods are used. Chemical method: the stain is prepared in 5% phenol, which has high affinity to mycolic acid. Physical method: use of steam during staining facilitates the entry of stain in the cell. The mechanism is based on phenomenon of relative solubilities of substances. The primary stain ZnCF is more soluble in phenol than in water or alcohol, and phenol is more soluble in mycolic acid, the waxy substance in cell wall of acid-fast organisms. Hence, cell get colored by ZnCFs during steaming process, which softens the waxy cell wall. Once the acid-fast bacteria gets stained, it does not decolorize even by strong decolorizing agents, as phenolic stain is more soluble in mycolic acid than in acid alcohol and appear pink in color. The non-acid-fast bacteria do not contain mycolic acid in their cell wall. The ZnCFs get decolorized easily and get stained by counterstain, that is 0.3% methylene blue or 1% malachite green, and appear green or blue in color. Observation: under oil immersion lens, acid-fast organisms will appear red in color, while non-acid-fast organisms will appear blue or green in color. Applications: detection of Mycobacterium tuberculosis from sputum sample, detection of Mycobacterium leprae in the smear of skin lesions, nasal discharge or tissue sample. Thank you.